drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Thank you for joining me.
Oh, thank you so much for having me, Nick.
Do you want to start off by just telling everyone a little bit about who you are and what your lab studies?
So I am a geneticist. Well, I train as a geneticist, but I'm a professor of molecular neuroendocrinology here at the University of Cambridge, in the UK, where we study two things. We study the genetics of body weight. On one end of the spectrum happens to sit obesity, and how the brain controls food intake. So those are the two elements that we study.
And when we think about the genetics of, obesity, metabolism, stuff related to that, probably the most famous place people often start is this famous mouse model that had a mutation that resulted in extreme obesity. Can you start us off by just talking about that mouse model? What is the phenotype and what is the genotype there?
So the phenotype, the mouse model was called the obese mouse, lacking because the mouse was obese. So a disappointing failure of imagination. Pardon me, a disappointing failure of imagination. A naturally occurring mouse at the Jackson Labs in Maine, and identified actually in the late 40s. And so it took a while. So people were studying this mouse. It was called the obese mouse for that reason. It was severely obese. It was in homozygous state. It was infertile. It was a recessive disorder, so that, in other words, the heterozygous mice were of normal weight. But it was only in the mid 1994, actually, so the early 90s, that the Friedman lab at the Rockefeller University in New York, in the age of molecular biology, identified the mutation causing this obesity, and that was a mutation in the gene called leptin. And so the OBOB mouse, and this was in effect the first real molecular handle that we within the whole community got, that body weight was not simply a matter of willpower, shall we say. So this mouse lacked a hormone, leptin, which is produced from fat. We'll come back to that, but lacked the hormone leptin, and as a result was severely obese. And from there came the whole concept that we sort of work on today with regards to body weight regulation and feeding regulation, and that of circulating hormones in the body that reflects your nutritional status, either long-term from fat or short-term from your gastrointestinal tract, that sends signal to the brain, and your brain then responds, translates, and integrates these long and short-term signals, and then influences your next interaction with a menu, with a refrigerator, or with a restaurant. Mm-hmm. So that, in essence, is that OBOB mouse, the obese mouse.
So this mouse is homozygous for recessive mutation in what we eventually figure out is the leptin gene, the gene encoding leptin, which is an important hormone we'll discuss. This mouse is severely obese. So if you look at pictures online,
they're spherical.
Yes,
it's a spherical. It looks like a tennis ball, except
yes,
it has whiskers on one end and tail in the other.
So severely obese. And there's other phenotypes that maybe we'll get to at some point. You mentioned one of them, which is infertility. So, it's not just adiposity where something is happening here. There's other things going on, it appears. Was this mouse the way that we discovered leptin, or did we already know about leptin in some capacity?
No, we didn't know about leptin. So this mouse was the way we discovered leptin. Now, with the OBOB mouse, and actually it had a phenocopy. So there was another mouse called the DBDB mouse, or the diabetes mouse. That was another naturally occurring mouse. This was identified in the early 50s, in which people used non-molecular tools to try and work out what the hell was going on. Why were these mice severely obese? Okay, so the OB mice was obese mouse. The DB mouse was a diabetic mouse. But in effect, they were phenocopies of each other. They were severely obese. And what happened was scientists were doing these weird experiments called parabiosis. I don't even know what a parabiosis experiment is, but parabiosis is a reverse Siamese twin operation where what you do is and not allowed to do today, by the way, in today's climate of ethics and stuff, but in the 50s or 60s. And so what scientists did, was to sort of merge the circulations of mice that were severely obese, OB mice with normal mice, DB mice, diabetic mice with normal mice, and then OB and DB mice. And what they found was that if you actually stuck an OB mouse to a skinny normal mouse, the OB mouse lost weight. Okay. And then when they stuck a DB mouse, a diabetes mouse, to a normal sized mouse, the normal sized mouse stopped eating and died. Okay.
So something in the circulation is traveling between them. Is basically what this means.
Exactly. And then they did the same experiment with the OB and the DB. And ultimately, this then had the hypothesis that there was a circulating hormone that was missing in the OB mouse and a receptor that was missing in the DB mouse. And, it's a real stretch. But it turned out to be true because the OB mouse turned out to have a mutation in leptin, okay, which is the hormone, whereas the DB mouse turned out to have a mutation in leptin receptor, okay, which is the receptor. And it was only in the age of molecular biology that they then found out what those were. So the concept of a circulating hormone and a receptor was floating about.
Yes.
The identity of it wasn't until 1994.
I see. So people had these ideas, but it wasn't until certain tools were developed that you could actually figure out, okay, this is a circulating hormone and a receptor. So this leptin mouse has mutation in the leptin gene encoding the actual leptin hormone itself, as opposed to the receptor for leptin. But so you get this obesity phenotype, this infertility phenotype, really striking phenotype. Where is the leptin receptor concentrated in the brain and body?
So as it turns out, the leptin receptor is really quite complicated, and we know really a lot about one subtype of it, which is the signaling form of it, what we call the long form of the leptin receptor, and that the leptin receptor in this long form is a typical tyrosine kinase receptor. Okay, so you bind it, and then there's phosphorylation of tyrosine kinases in the cytoplasm, inside the cell. And but there are a whole bunch of other shorter forms of leptin receptors, and some even say circulating forms of leptin receptors that we actually have no idea what they do. And so the circulating and short forms are present in many, many parts of the body. The long form, the one with the tyrosine kinase receptors, are primarily neuronal within the brain.
I see.
And so they're everywhere in the brain. They're concentrated in the hypothalamus of the brain. Which is, for those of you who don't know where that is, the bridge of your nose, base of the brain, and it sits right above the pituitary, and that's where it tends to be concentrated, but it's expressed throughout the brain, the leptin receptor.
And so when you say that, so there's multiple versions of the leptin receptor, are these isoforms of the same gene or?
Isoforms of the same gene. Splicing, these are all splicing variations.
I see. So one gene can create multiple versions of the leptin receptor, but there's one form that predominates, or that we at least know the most about. It's found in multiple places, but it's found in the brain, concentrated in the hypothalamus, but located in many, many parts of the brain. And it's a particular type of receptor, and obviously this is what leptin binds to. So something about leptin's ability to bind this receptor is what's disrupted in that OB mouse.
The OB mouse had a mutation in leptin, which in effect stops it from binding. Yeah, correct.
I see. Okay, so the end result, so when leptin can't bind effectively to the leptin receptor, the result is this dramatic obesity phenotype. This receptor is located in the brain, in the hypothalamus, which was already known at that time to be crucial for things like feeding and metabolism in different ways. So it starts to point us to leptin's action in the hypothalamus. Before we get there, literally, I want to give people a basic sense for the hypothalamus in mammals. So you mentioned where it's located. It's essentially in the middle of our head, right behind our eyes, in sort of the center, right above the pituitary. Can you give us a sense for sort of the size and structure of the hypothalamus down to the cellular level as opposed to other parts of the brain here? So I'm thinking of something like the cerebral cortex. How does the basic architecture of that part of the brain differ from this part of the brain?
So the hypothalamus, okay, size first. In humans, anyway, right? In mice, it's a lot smaller. Clearly, in humans, it is about the size of your thumbnail and is shaped like an almond.
Hmm.
Okay, and it plays a role in terms of its role. It plays a role in all forms of homeostasis, so balance. It includes energy balance, which is what we're talking about, body weight balance. But it plays a role in salt balance, temperature balance, stress, sexual, reproduction, circadian rhythms. It plays a role in a number of different things that your body has to keep your body in balance. So homeostasis. But we're talking about body weight today, obviously. Now, as a part of the brain, it is it's not the most ancient part of the brain. That's the brainstem, but it's one of the most ancient parts of the brain, and it is definitely the most complex part of the brain. Okay, because what happens is it is formed of these groups of cells called nuclei. So as opposed to the nuclei within a cell, they're sort of nuclei of cells, shall we say, within hypothalamus. And it is the most heterogeneous part of the brain in terms of the number of cell types that are there. That's the first thing. The second thing is that it bristles with receptors because it acts as because of where it sits at the base of the brain, it senses circulating hormones, nutritional cues, which is what we're talking about, but a whole bunch of other things, stress hormones, sex hormones, all these hormones as well. So it's bristling with the receptors. It is also the part of the brain that secretes the most hormones, neurohormones, and neuropeptides. Mm-hmm. So with all that in place, if you go to some other part of the brain, the only part of the brain that perhaps is sort of similar to it is probably the hindbrain, mm-hmm. but that also sits sort of as a sensor area. The rest of the brain is complex. Look, it is very complex, okay, particularly the higher order, but they're not as heterogeneous. They're not necessarily organized into nuclei in as such, certainly not as many as they are within the hypothalamus. Mm-hmm, so that really is it. It's the most complex part of the brain, mm-hmm. undoubtedly.
Yeah. So to reiterate some of that, so it's an ancient part of the brain. So the hypothalamus exists basically in all animals, and it's going to have mm-hmm. similar characteristics across all of them. There's going to be differences as well, and we might talk about some of the human differences compared to rodents, say, but the basic features of the hypothalamus have been around for a very, very long time.
Yeah, they're sort of versions of some of the neuronal types within the hypothalamus that exist all the way down to worms.
Wow.
The worms don't have a hypothalamus per se, mm-hmm. but the sort of proto types of these neurons were already there in C. elegans in the worm.
Yeah. And so you said in a human, it's roughly the size of a thumbnail. It's located sort of in the center base of the brain. So the cerebral cortex, right? If you just think about it, the wrinkly outer part of our brain, it's much, much bigger. And you also said, so it's evolutionarily old. There's going to be a lot of conserved ancient functionality here that's common across animals. It's got an interesting architecture at the cellular level. So you said there's more cell types here. There's more cell type diversity here than probably any part of the brain other than maybe the hindbrain. So if we were to look at, say, the cerebral cortex and sort of count or use molecular techniques to identify distinct types of cells, there's way more diversity in the hypothalamus than other parts of the brain. And based on where it's positioned and based on some other things we're going to talk about, it's connected to a lot of parts of the brain, and it's also sort of a sensor integrator of signals, not just from other neurons, but as we sort of maybe hinted at the beginning, things like hormone signals in the blood as well.
Absolutely. So it acts as a sensor for many hormones within the context of body weight. It's probably safe to say it's a fuel sensor within the context of body weight. Yeah. Mm-hmm.
And so talk a little bit about this hormone sensing capability and so normally when we talk about the brain, we talk about the blood-brain barrier and how it exists and how it's preventing just anything from getting into the brain from the bloodstream. Nonetheless, there are parts of the brain where neurons can sample pretty much directly from the bloodstream, or at least the blood-brain barrier is weakened compared to other parts of the brain. Can you talk about that a little bit with respect to the hypothalamus?
So the hypothalamus and the hindbrain actually are both what we call circumventricular organs. And that means that they It's not weak. I think it's the designs rather
Hmm.
than a weak in which they are fenestrated. Okay, so in other words, there is the ability of peripheral hormones to make it into the brain. This is not. It doesn't go through by diffusion. I want to point out. All right,
Hmm.
it is a regulated process
Got it.
in which in these parts of the brain they're able these hormones and many other things, including nutrients themselves, manage to actually get across this layer and into the hypothalamus and in the hindbrain as well, where the hypothalamus therefore samples and then is able to use the information for command and control.
Mhm. So hormones and nutrients can literally get into the hypothalamus. It's not a passive process. This is supposed to happen, so to speak. It's designed or evolved this way. Um, and we talked about leptin
Evolve,
before.
that's right, not design. Yeah,
Yeah,
evolve in
evolved,
this way. I'm not
designed
a
designed
just to
through a
be clear.
distributed,
Yeah,
decentralized natural process.
yeah.
Um, so hormones obviously can get into the hypothalamus. Leptin is what we talked about at the beginning. What talk about how is leptin, well, actually before we talk about what leptin is doing in the hypothalamus, where is it actually coming from? We know it gets to the hypothalamus from the bloodstream, but where's all of this leptin coming from originally?
So leptin is produced exclusively from fat, adipose tissue, and specifically the adipocytes. So in other words, fat obviously has other stuff in it, but primarily are adipocytes. And so leptin is produced exclusively from adipocytes, and it circulates in proportion to the amount of fat that we have. So the more fat you have, the more leptin you have,
Hmm.
and the
so,
less fat you have, the less leptin you have.
so it's secreted from fat cells, and the more of them you have, the more leptin that's circulating.
Hmm.
Okay, so it circulates, it gets up to the hypothalamus, it can get in there because this is a circumventricular organ, and certain hormones and other things can get into the hypothalamus. What's sort of going on next? What are the main cells or parts of the hypothalamus that are sensing leptin? And at a high level, what is a high level of leptin actually signaling to the brain, do we think?
So a high level of leptin is signaling to the brain that you have a lot of fat, and a low level of leptin is signaling to your brain you don't have a lot of fat. But it acts throughout the entire spectrum. Okay,
Mm-hmm.
so in other words, so you obviously it's a full spectrum because there's a concentration gradient. Now, what happens when it actually gets to the hypothalamus? And this is and to be fair, I have to oversimplify it because it's just too complex and we don't know all of it.
Mm-hmm.
But when it gets to the brain, one of the things it does is it signals to a population of neurons called POMC neurons, pro-opiomelanocortin neurons, at the arcuate nucleus of the hypothalamus, right at the very base. And then when leptin signals to leptin receptor expressing POMC neurons, POMC is then sort of post-translationally modified, sort of chopped up, okay, by prohormone convertases. And then they in turn project to and signal to another part of the hypothalamus called the periventricular nucleus, which is another part of the hypothalamus, where melanocortin receptors exist. And because this melanocortin make melanocortin peptides, which bind to melanocortin receptors. Now, this whole pathway, leptin receptor, melanocortin receptors, is called the leptin-melanocortin pathway because it's the namesake or
Mm-hmm.
the central leptin-melanocortin pathway within the brain. And put simply, this pathway acts as the fat sensor within our body. So your brain senses the amount of fat, and this is the key. This is the key pathway within our brain that senses how much fat we have. It's conserved. All mammals have this pathway, and most higher vertebrates who have a demonstrable fat pad will have a demonstrable amount of fat that you can see
Mm-hmm.
and weigh will have this pathway.
Mm-hmm. So leptin binds to leptin receptors in the hypothalamus. It can get in there because the hypothalamus is a circumventricular organ and is essentially built to sample the bloodstream. One of the main targets within the hypothalamus are these POMC neurons, and this is part of the leptin-melanocortin pathway. So basically, the OB mouse that you told us about at the beginning, it had a mutation in the leptin gene, and it was severely obese. It was basically unable. That mouse was unable to sense its own body fat level.
Correct.
Yes.
Yeah. The signal between fat and brain broken. So the brain, in effect, thinks it has no fat,
Mm.
broadly speaking, because it can't sense the leptin. It says, "Oh shit, there's no fat. I've got no fat," which is a bad thing,
Yes.
because we're designed to have some fat in us.
Hmm. So is it, so how does this hook into other parts of this region of the hypothalamus. Is it fair? So this is a fat sensor, and obviously this is going to tie into things like hunger and food intake. But do people think of this as like a satiety controlling circuit, or how do we think about that?
That's an interesting question. I think when it was first discovered, leptin, I want to say this conserved, so it's for all this is found in humans as well. But when it was first discovered, it was said to be a satiety hormone. I don't think it is a satiety hormone. It is a fat hormone. It is a how much fat do you have hormone.
Mm-hmm.
Okay. But the crucial thing is it's designed to signal not overnutrition because too much fat, because from an evolutionary perspective, there was no pressure there. We never had too much. The vast majority of evolution, we never had too much food where we had to design an entire system to tell
That
our
was
brain
never
we
an
had too much
issue
fat.
until, blink of an eye ago.
Over 30, exactly. Literally a blink of an eye ago, like 30 years or 40 years or so. It's designed to let your brain know you have not enough fat, which means that you're
Mm-hmm.
starving in effect.
Yes.
Okay.
And just to
So it is a
emphasize it for
starvation
people,
hormone.
just to emphasize it for people, right? Not enough fat for basically all of evolutionary history of animals was a very big, very frequent problem. Too much fat was essentially never a problem. And so the system evolved to reflect the fact that no fat was a problem and too much fat essentially never was.
Exactly. And so there was no pressure. There is a pressure on total size, and we can get
Mm-hmm.
to that maybe. And that was probably predator wise, so you need to make sure you don't get too big. It's sort of like the peacock, right? The peacock can with this silly tail. This is probably the maximum size it gets before it gets eaten. And so we do probably have a maximum size, but undoubtedly the key selection pressure is starvation, having
Mm-hmm.
not enough fat.
Mm-hmm. Okay, and then so you said these POMC neurons are a key type of neuron in the hypothalamus that are sensing the leptin because they have the leptin receptor. Pro-opiomelanocortin. This is a peptide as opposed to say a neurotransmitter. Can you talk a little bit about the difference between peptides and neurotransmitters and the significance of having that these neurons have peptides and what that sort of enables them to maybe do?
Okay, so when we think about the brain, there are two different ways that I'm simplifying, but there are two different ways that communicate. There is the wired connection, and the wired connection, which means that synapses are going and going to another synapse. And broadly speaking, they communicate through these things called neurotransmitters and electrical impulses. All right. And so these include things like GABA, it includes things like glutamate, it includes acetylcholine, a number of these transmitters that actually move stuff, but it's wired. Okay. So they come out actually at the end of the synaptic boutons, which are projecting to soma with the cell bodies. Now, the thing about neuropeptides, however, is it's almost a wireless connection because it's hormonal. It's neurohormonal, but it's hormonal, which means that they can be released from the synapse, yes, but they can equally also be released from the cell body. Like in other cells in our body where there's a vesicle, it comes to the surface and it releases a hormone. The same thing can happen within the brain. And so it sort of can diffuse is a very, very strong word here.
Yeah.
But the rate of signaling is very different, and kinetically is very
Yes.
different because rather than really quick microsecond impulses, you have to wait for the hormone to be released, move about, get to another cell. So that's the difference between the wire. One is wired, which is the neurotransmitter, and one is
Mm-hmm.
wireless.
So there's differences in the spatial and temporal dynamics of these different communication modalities. When you're talking about neurotransmitters and wired communication, it's like very fast, millisecond to millisecond, point to point communication between the wires, which are the neurons in this case. But when we're talking about these peptides that can be released from some neurons, it's a much sort of slower, lower resolution response, essentially.
It's still pretty quick, but
It's still
but
pretty
it
quick.
is but it is far log scales slower
Yeah.
than neurotransmitters. Yeah.
Mm-hmm. Mm-hmm. And I mean, is this how do you think about this? Is this related to the fact that these neurons are, say, sampling hormones from the blood and they don't merely need to do fast point to point communication, they need to do these sort of longer integrations of slower signals?
I don't think they're mutually exclusive to each other because I think probably when um And that's an interesting question, actually. Why there is these two? I think, okay, let's step back. I get the feeling that there are two almost separate, different separate types of ways that that your neurons communicate. Actually, there's probably a third type. We can get to that. Okay, that's the plasticity type as well. But with regards to sending stuff out, okay, I think this gives us more a finer ability to tweak the system. You need stuff that happens quickly. You need stuff that actually happens over a period of time, which I think this gives us the full sort of like spectrum of speeds of communication for different reasons. Okay, we need stuff that moves quickly so that we can, we can stop things, we can, and calcium channels can open and all kinds of things happen. But we also need things to tell us you have this amount of fat in your body. Okay, and that maybe is a longer term sensing process. I mean. This is not my area of expertise. Can I please just stress the difference between this? But this is what I think.
So leptin can activate these neurons. When these neurons become active or they're made active, how does it change their feeding behavior?
So there are two groups of these melanocortin. I neglected to mention another group of neurons. So when leptin comes up, maybe we should do this. It signals to POMC neurons, and POMC neurons are anorexigenic. Anorexia, not eating. So in other words, when POMC neurons are activated, you tend to eat less. Okay, so they're anorexigenic. Now there is a counterpart set of melanocortin neurons in the brain called the AGRP neurons, agouti-related peptide neurons. Okay, and it's a long story why they're called that, but they sit in the same part of the brain, the hypothalamus. They also express leptin receptors. But when they get activated, the AGRP neurons, they encourage food intake. They are orexigenic, which is the opposite to anorexigenic. And so what happens is both of them signal to the melanocortin receptors, and they act as a gas pedal and brake. All right. And it's the tone of how much of POMC signal versus AGRP signals on the melanocortin receptors that sort of feather how much you eat. It's a very, very, very subtly tunable system. So it's not on or off. It literally is the ratio of AGRP signal to POMC signal, which then sets how much we are likely to eat in any given meal.
So you've got two neural populations with basically opposing functions with respect to feeding. The AGRP neurons promote feeding. They're orexigenic, that's the technical term. And then the POMC neurons that are sensing leptin, they are anorexigenic. And so these two have similar targets, and sort of the balance of activity between the two is a big part of what controls hunger and feeding levels. I would imagine, other than, if the hypothalamus is this integrator of multiple signals, and obviously how hungry you should be, how much you should be eating, that's going to be related to how much fat is currently on your body, but it's going to be related to other things as well. I would imagine that these cells are sensing directly or indirectly other things rather than the fat content of the body, such as, say, nutrients that are related to a meal you may have just had or not had.
Yes. So they also have receptors for some of the gut hormones, for example. The GLP-1 receptors are present on POMC neurons, not all POMC neurons, but some POMC neurons, for example. They express the insulin receptor. Now, insulin, we understand, famously regulates our, as an intermediary, it's important in intermediary metabolism, particularly for carbohydrate absorption, but actually all fuel absorption. But insulin also reflects long-term fat mass and nutrients in of themselves. Okay, so for example, glucose sensing or fat sensing or protein sensing even. Okay, so not all of these neurons have all of these receptors, but all of these receptors are present to some degree on these neurons. So the neurons really sense an integrated, because when we eat or don't eat, it's a repertoire of hormones and nutrients and things that are released. And so it's designed to sort of dip a toe into the soup. And sense that.
Yeah. So this part of the hypothalamus, the hypothalamus is at the base of the brain, right above the brainstem, but the arcuate nucleus, where these cells we're talking about live, is at the base of the hypothalamus. So it really
Yeah.
is like, dipping a sensor into a stew and sort of detecting the full soup of what's in there.
Yeah, exactly.
Interesting. So let's give people a sense for how dramatic some of these effects are. So you said that you have these AGRP neurons, and they are basically they're opposing the action of POMC neurons, and they are orexigenic. So they promote feeding. What happens if you take a mouse that's got a full tummy, it just ate a full meal, and you turn on these AGRP neurons?
They eat immediately. And that's the thing. So like if you do it like using optogenetics, so something very, very quick, all right? Um, you can do it using optogenetics, which is quick. You can do it doing chemogenetics, which is like dreads and stuff, and they'll take a little bit longer. But ultimately, when you activate AGRP, when you inject AGRP, but when you activate AGRP, the mouse starts eating no matter how full they are.
Yeah, and the famous, to give people a sense here for how strong this effect is, the one of the kind of stats or observations you often hear is you take a fully fed mouse at the end of its night when they're normally eating, so they're nocturnal. This is, their tummy's full. This is when they would normally eat the least amount they're going to eat all day. If you turn on these neurons by one means or another, they will eat oftentimes as if they've been starved for 24 hours straight, which for a mouse is very, very hungry.
It is very, very hungry. It's really quite dramatic. Just one comment to make on the dynamics of POMC and AGRP, the differences. So unsurprisingly, when you take a mouse or a human being for that matter, but easier in a mouse, obviously, and you fast them, so in other words, you nutritionally perturb them, what happens is AGRP is very volatile. Okay, so in other words, levels of AGRP, mRNA at any rate, if you fast the mouse, will go up 8 to 10 fold in an overnight fast, for example. POMC, however, for whatever reason, okay, is far less volatile, far less dynamic. It probably changes twofold, maybe threefold in a fasting feeding type of situation, as opposed to AGRP. So of the two pairs, AGRP is definitely the more volatile of the siblings, shall we say?
And then what can you tell us about sort of the one of the next nodes in some of the circuitry? You said both of these cell types converge on the melanocortin receptor. You mentioned a place called the PBH. This is just another part, sort of the next part in a sense of the hypothalamus. What so this nucleus of the hypothalamus where the POMC neurons and the AGRP neurons live, it's it's a sensor dipped into the stew of the bloodstream. It's sensing leptin, it's sensing other nutrient content in the bloodstream, and then there's some net signal that gets sent out, some amount and pattern of activity from the POMC part of it, some amount from the AGRP or go eat part of it, and these signals are converging on melanocortin neurons elsewhere. What is sort of the next step in the circuit? What's happening there?
I think the melanocortin neurons are probably the first of the effector neurons.
Mm-hmm.
Okay, so they're one step away from the sensor, which you can, I mean, it's POMC and other neurons, they're the sensors. And then obviously they then begin to, something has to happen, right? Eventually it's got to get to our eyes, our nose. My hand has got to reach a spoon and dip it into the ice cream bowl and put it in my mouth. So I think the MC4 receptor is the first of these effector neurons. And so they then sense either the balance between POMC and AGRP and then begin to influence feeding behavior.
Mm-hmm. So roughly speaking, we can think of the arcuate and the POMC and the AGRP neurons as sensors of
Yes.
Fat content, nutrient content in the blood, et cetera. And the sort of net or integrated signal from them that converges on melanocortin neurons at the next node, that signal is basically saying this is our need to feed, and then that's going to then be broadcast to other systems in the brain ultimately that are going to tell us to pay attention to certain things with our eyeballs or move our body in a certain way, et cetera, et cetera.
Correct, because now the POMC is an AGRP, both are only expressed in the arcuate nucleus of the hypothalamus. POMC in the hindbrain as well, but we'll ignore that for a second. It is only there, and there are very few of them. In the mouse, there are probably only 3,000 POMC neurons and an equivalent number of AGRP neurons, like literally, all right? In humans, there's probably tens of thousands of them, but we have hundreds of billions of neurons in our brain. So these are very, very few. The moment you get to the MC4 receptor as a secondary, second order neuron, shall we say, effector, There are far more of those. And the MC4 receptor, I mentioned the paraventricular hypothalamus, but it's actually present in a wider spectrum of neurons within the brain. There are far more MC4 receptors than there are POMC neurons.
So these are fair to say that, so these neurons they're tiny in number in the context of the full brain, and they're pretty dedicated. It's fair to say probably that they're specialized for a particular type of signal.
They are. So POMC, broadly speaking, they're specialized for energy homeostasis. I mean, it does a number of different things as well in terms of glucose and stuff, but broadly speaking, it is specialized. They're nutrient specialists.
And are these neurons, I mean, are they just affecting feeding behavior and hunger, or are they also affecting, is their activity affecting other aspects of metabolism, like your basal metabolic rate and things like this?
So now that's an interesting, from the mouse perspective, there is evidence that perturbation of the leptin-melanocortin pathway has changes in terms of metabolism, either at basal level or reactively in terms of for other things you might be doing, say changing your diet to a high-fat diet or something like that. Humans, however, with these same mutations and hence have also severe obesity, there has been no evidence to date of any significant changes in metabolism when it comes to mutations within the same pathway. The question comes down to why. I don't know. I think there is going to be a size issue where mice are clearly far tinier. And so I just get the feeling their metabolism is very twitchy. Human beings are large, lumbering creatures. Maybe there's an issue where it's very difficult to finally measure metabolism in humans. But these studies have been done in these big chamber calorimeters. They've been done pretty well. So it's interesting. So there are changes in metabolism in rodents. In humans, far less so. In humans, the only genes so far that have shown significant changes in metabolism are changes in the thyroid hormone system. But with regards to the leptin-melanocortin pathway, severe obesity, yes. Okay, eating a lot, yes. Metabolism, no. Actually.
Got it. Okay, interesting. So let's go back. So now we sort of have some sense of certain parts of the hypothalamus. We've got the circumventricular organ, the hypothalamus itself, and the arcuate nucleus in particular. It can sample from the bloodstream. It can detect leptin and other things. You've got your POMC, anorexigenic neurons. You've got your HRP, orexigenic neurons. They've got, it's your pedal and your brake with respect to feeding behavior. They send signals downstream. We talked about this OB mouse at the beginning that was severely comically obese because it's got a mutation in leptin. So its brain is basically fooled into thinking it's got no body fat because it can't detect that body fat signal that's circulating in the blood. And therefore, the animals are hyperphagic. They're eating a lot. Have we observed that mutation, the equivalent mutation in humans, and does it have the same result?
Yeah, so there are mutations in leptin and the leptin receptor. We'll start with those two in humans as well. And they are in effect phenocopies. The only phenotype that doesn't come out is the metabolism thing, which I talked about. Everything else is true. So obviously they have either hyperphagic, hyper for more, phagic for eat, and it's a pathogenic term. Okay, so in other words, they have this really voracious appetite because they think they're starving. Okay, they have a brain that thinks they're starving. They are infertile, or at least they have something. Humans have something called hypogonadotropic hypogonadism. So infertile. They don't go into puberty. They also have an impaired T-cell mediated immunity. A bit odd to be talking about within a obesity setting, but actually these last two phenotypes, together with the starvation, together with the fact that leptin reflects fat mass, begins to give us an idea of the biology of leptin. And the reason there's infertility and a weird immune system, it's because when you are starving, a number of things happen. Okay, when you're starving, you obviously have to eat.
Yes.
And when you're starving, you eat foods that you don't eat poison that will kill you, but you eat foods that you may not find palatable.
Yeah.
So these kids, for example,
you'll be less discriminating in what you choose.
Yeah, you're not discriminating. So these kids, for example, have to have locks on their freezers because they'll go in and eat frozen food.
Yeah.
So it's clearly a pathology, but it's not poisonous. It's
Yep.
just unpalatable if we're not starving.
Right,
But
right.
if you're starving, you'll do
Mhm.
it.
So, from an evolutionary perspective, the basic idea here is if your brain truly thinks it's starving and you don't have any fat because you can't sense this leptin hormone, you will, from an evolutionary perspective, right? It's all about getting your lineage to persist, which means you've got to reproduce, which means you've got to survive long enough in order to reproduce. And so if you are about to starve, obviously that's not going to happen. And so the body deprioritizes A, fertility related things, and B, fighting off infections so that it can sort of essentially allocate resources to not immediately dying through starvation. Is that the basic sort of
Triage.
idea?
It's doing triage.
Yeah.
So your brain says, all right, guys, we just have to triage this and begin to triage calories for a brain. And like specifically glucose. Your brain, for those of you who don't who just dig back into basic biochemistry, your brain doesn't use fat. Okay, your brain uses glucose and some ketones at moments of stress, but your brain doesn't use fat. So as a result, you really have to maintain your glucose levels above a certain level for your brain to function. And so it begins to triage all kinds of things to ensure that the command and control center of the brain continues receiving glucose.
Mm-hmm. So we have comparable mutations in the leptin gene in humans that we see in this OB mouse. They have, you said it, phenocopies. So they are very obese, and you just described how they have this voracious pathological hunger. I'm guessing this is a very rare mutation.
Yeah. So in humans, so it was first so the people who identified the first leptin mutations were he just retired as head of the department, but my head of department for a long time, Steve Ratley, and his at the time clinical fellow Sadaf Faruqi, and they identified the first kids with leptin deficiency in 1997. Now, since 1997, there've probably only been less than 100 families worldwide with mutations within leptin. Further up the pathway is very different, but within leptin and maybe an equal number of patients with leptin receptor mutations, roughly. So incredibly rare, by no means a common cause of obesity.
Mm-hmm, mm-hmm. So the discovery of this mutation, it was a significant mutation because it led to all this sort of path of discovery that we've started to discuss a little bit. But certainly this one mutation isn't going to explain something like the obesity epidemic that humans are experiencing, because most obese people don't have this particular mutation. I want to ask you now a little bit more about genetics as it relates to obesity and adiposity. What can you start to tell us about the genetics there and other mutations and other genes that we think are linked to one's propensity toward obesity and high fat deposition? They might not be as dramatic as this leptin receptor mutation, but are there other genes or a strong genetic component here that we should know about?
So there, I guess you could almost think about it in two different ways. There are very extreme cases of obesity, and there's more subtle changes like normal distribution of body weight. Sticking with the extreme obesity cases, they eventually overlap. But speaking about the extreme obesity cases, the vast majority of single gene or at least very, very strong genetic drivers for obesity lie within this leptin-melanocortin pathway. Throughout the entire pathway.
Mm-hmm.
All right, mutations in all the genes result in severe obesity. There are other genes to be found and have been identified, but in total, if you actually add those up that are not part of the leptin-melanocortin pathway, they are tiny, itsy bitsy, teeny weeny things. And what they
Yep.
do is they give us insights into certain other biology, but the primary genetic driver for it is variation within the leptin
Mm-hmm.
melanocortin
So,
pathway.
so if you sort of break or disturb a number of different levels of function in this leptin melanocortin pathway, you can get severe obesity phenotypes. It's what you're basically saying is this leptin melanocortin pathway is sort of like this is the important piece. It's not that there's nothing else going on elsewhere, but that this is a key node, and you can break it in multiple different ways.
So it's interesting actually. There's two ways of viewing it. It's sort of, remember the memes which go about which show you the picture of the airplane being shot up in World War II,
Yes, yes.
and what they show. So there is a survivor bias.
Yep.
Okay, so I think it's definitely a pathway that is tunable and tweakable,
Mm-hmm.
and therefore is very important. I just get the feeling it's probably the reason we see so many mutations is because it's a pathway that can take a lot of hits and it's a tunable system. I get the feeling that the actual truly existential pathways, if mutated, we're dead. And
Yeah,
so hence we don't see mutations in those very, very
I
frequently
see.
because they probably do not have any redundancy in those particular pathways, or at least are not designed as a rheostat. They're not designed to be tuned.
Yeah.
And so if you knock them out, that's it. So the importance, it's an interesting discussion, probably too
Mhm.
niche for people, but I don't know if it's the most important pathway. It's certainly the most
It's very
tunable
tunable.
pathway.
I see.
Yeah,
I see.
yeah.
Okay. And so I want to ask you a little bit more about genetics before we circle back to neurobiology. Um, so there's this sort of just general question in society. So obviously we've got this obesity epidemic. Everybody's got a different opinion about exactly how we explain that and what to do about it. On the genetic side, so obviously there's not a bunch of people walking around with OB mutations in the leptin gene. It's not like in 1940 there was a mutation in the leptin receptor and it somehow spread, and that explains the obesity epidemic. But I want to sort of ask you how much of the variation we see in body weight and obesity can be attributed to genetic factors. And we'll probably have to spend a little bit of time here really unpacking that for people because it can be confusing to think about. But I'm just going to start there at a very basic level. How do we start to think about how much of the variation in obesity is attributable to our genes as opposed to our environment?
So we do have a number for that actually, and that's because of studies from twins. Okay, you can do the same thing for any given human trait or behavior. And a lot of it has come from twin studies. You have clearly monozygotic twins, identical twins, who are for all intents and purposes genetic clones of each other. And then you have dizygotic twins, so non-identical twins, and they would share as much genetic material as you would with your own siblings or your parents, so 50%. And so if you study a large enough cohort of twins, tens of thousands of pairs of twins, identical and non-identical, then you can ask a question about any given human trait or behavior.
Mm-hmm.
What happens when you share 100% of genetic material versus 50%?
Mm-hmm.
And from there, you can then come up with a measure called heritability, which is what you just asked. The variation in a given trait that is down to genes versus non-genes, which we can say environment. Okay. And if you do that math, what you end up with is the heritability of fat mass, body weight, okay, is between 40 to 70%. So it's a pretty big range.
Mm-hmm.
It's a pretty big range. But broadly speaking, that is the amount that is attributable to genetic factors.
Mm-hmm. And so let's really unpack that for people who are unfamiliar with this stuff. So you said 40 to 70%. Let's just I'm going to use 50%, a number kind of in the middle of that range.
Mm-hmm.
Does that mean that 50% of the fat on someone's body is due to their genetics only?
No. So heritability is a population level measure. So what you got to do is you got to look at tens of thousands, hundreds of thousands, millions of people and say that at the population level, you have variation in given traits. And of that, it is likely that 50% on average is down to your genetics versus the environment. As an individual, clearly your body weight is what your body weight is. And you're then asking a different question. You're asking a different question about the role that genes play within an individual. But heritability is a population level measure.
Yeah. So we're talking about the variation between people in a population. This can be a little subtle for people, but can you give us a sense here for what the variation in like BMI is or obesity? If we were to take random people from the population, how variable is it actually?
It's very variable because obviously you go so actually the distribution is one of a tail. Okay, so you sort of have it's not a normal distribution per se. It's like a distribution with a long tail, and the long tail is where the body weight is. Okay, because you got people who are, most of us are average body weight. Okay, a little bit heavier than we like. And then as we get out to the tails of severe obesity, then there are fewer and fewer individuals. But it's sort of like a normal distribution with a very long tail on the right hand side. But the variation is enormous because we can go around and we can see that they're really super uber thin people for many different reasons, okay, but uber super thin people. And then you have the rare cases of severe obesity in which people need to, get holes cut out their roof and craned out of their house, like really severe type of obesity. But most of us clearly sit within a normal range, even though this range is increasing as we speak.
Yeah. So depending on how you count, yeah, I mean, there's no start date for the obesity epidemic, but in the mid 20th century, let's say 1950, give or take, obviously obesity rates were very different. So the absolute number of obese people was smaller then than it is today, and the distribution of fat phenotypes was narrower. How do population level changes in rates of obesity like that, both the average level and the distribution, does that affect our estimates of heritability at all over time? If you were to estimate the heritability back in 1950 versus today, would it be affected by those changes?
No, because the heritability is not of obesity. It's the heritability of body weight. And so hence, yeah, okay, the dynamic range may be slightly narrower. So maybe you need a larger number of twins, for example, in order to actually identify the heritability. But the heritability is the heritability, right? And so you're just looking at body weight size, and with enough pairs of twins and enough people, then you can actually see that. So it just narrows the dynamic range slightly. It's not going to change the heritability. What will change the heritability, however, interestingly enough, is the environment. Because heritability is a it's not you can't just study genes in isolation because your genes are not there to function in isolation. Your genes are there to allow you to adapt to the environment. And so if the environment changes, it can change the heritability of your of what happens. So this 40 to 70%, why this huge gigantic range? Why didn't I say 50%? Why didn't I take an average? Because the environment really influences the level of heritability.
That's something I want to emphasize for people because this can trip people up. There's this inextricable link between genes and environment. Like again, you just said the environment itself, changes in the environment change the heritability of these things.
So let me give you one example, all right? Because it blew my mind. Okay, so a colleague of mine, Professor Claire Llewellyn, who's based down in London at University College in London, she has a twin cohort. Okay, and she is interested in body weight as I am, which is why we're colleagues. Now, the interesting thing is that her set of twins, it's called the Gemini study, for those of you who want to Google it, Gemini. Her set of twins, she includes all the anthropometric measures that you would assume would be there, but she also has, interestingly, socioeconomic markers for these twins. Okay, so within society. Now, if you go, or if she went to the twins in our households, okay, sort of a academic professional class individual, not someone who's rich, just we don't have to worry about our food, right? We know where we are, type of situation, middle class, but in the UK terms, then heritability of body weight was around the 40% mark. Okay. Now, when you went to the households with the poorest food environment, the lowest socioeconomic status, suddenly heritability maxed out at 70%. I read the study, I was going, dude, what is going on? And this is the way that I rationalize this particular concept. All right. Now imagine. So I accept I'm a privileged human being, right? I'm a professor at Cambridge. I live in a nice leafy village. Now, my nice leafy village here, Deliveroo is so far away from anything. Deliveroo doesn't deliver out here. Okay, and we don't have any takeouts in our village. So imagine me, and I have a high genetic drive to eat. Just as an example. Now, if I were hungry tonight, for example, I can't just wander around. I can't call Deliveroo. I have to look at my fridge or get in my car and drive to the supermarket and do something about it. Okay. But if I look at my fridge, maybe I'll find carrots. Maybe I'll find some hummus. Now, imagine there's a me in terms of the same genetic drive to eat, but I'm now in town. Okay, I'm now in a inner city location. I live above a takeout. I'm in an area, maybe a food deserty type place. Deliveroo definitely delivers. All right, and suddenly my ability to fulfill that drive is far, far, far easier. Okay, I'm getting in my car. I'm going, oh, I'll wait till tomorrow. Like, really, I can't be bothered. Whereas if I can run down the stairs or call Deliveroo and eat it, I probably would. And so that's the difference, right?
Hmm. Hmm.
I'm privileged. I live in a leafy green place. Whereas if I was less privileged and lived in not a leafy green place, but had the exact same genetic drive to eat, then I would be able to respond to those signals better.
Right, right.
You see what I mean?
Yes, yes. Essentially, the extent to which you've got a genetically driven propensity to hunger, whether or not that can lead to an outcome of overeating is going to depend on the availability of the food in your immediate environment. So, I mean,
And the
even at
type
the
and
yeah,
the type of food.
yeah, and the type of food. Like even at an individual level, this is this is actually like kind of how I regulate myself in my home. I know that what I eat and how much I eat is going to be tied to what I have in the cupboard. And so I make myself not have full cupboards most of the time because I know that if it is full, I'm going to eat way more just because I can.
Exactly. And that's just a situation. And then there's the whole complexity of what you have in your cupboard. Those tend to be more processed foods because they are in your cupboard rather than in the fridge, and therefore longer shelf lives. And they tend to be cheaper than fresh food, right?
Mhm.
So you now then go down the rabbit warren of the fact that, well, okay, so not only is socioeconomic status linked to where you live and what you can access, then what type of foods you have. Are you eating fresh food? Are you eating food that is pre-processed? And then you can get into a deep discussion about socioeconomic class and health and body weight.
Mhm. Mhm. Okay, so I want to talk about glucagon like peptide 1. So you mentioned it briefly
Hmm.
earlier. I want to talk about what that does sort of normally, and then we'll talk about the new weight loss drugs. So, what is glucagon like peptide 1, and how does it work and tie into some of the circuitry and stuff that we were talking about earlier?
Okay, so glucagon like peptide 1 or GLP-1, otherwise we'll be here all day, okay, is what we call first of all, it's a gut hormone. So it's one of the hormones that go up. We know of 20 different gut hormones. So GLP-1 is one of them. It is called an incretin hormone. Now, there are two incretin hormones, GLP-1 and GIP, which I'll get back to in a second. Now, what are incretin hormones? I guess let's deal with that first. So when you inject yourself with glucose, sugar, your insulin levels go up. We understand the biology, obviously. But when you eat glucose, and so hence has to go through the stomach and then passes through the gut wall, the act of eating it enhances the insulin secretion by 8 to 10 fold. Okay, exactly the same amount of glucose.
Mm.
Now, this effect is called the incretin effect. And the reason it happens is because as glucose passes through the gut wall, GLP-1 and GIP in particular go up. Okay. And they signal to the pancreas and enhance insulin secretion. So GLP-1 and GIP are incretins because they enhance insulin secretion when you eat sugar as opposed to inject sugar.
I see.
So
So,
that's first.
so,
Sorry,
so
go.
because we're animals that evolved and we eat through our mouth and swallow it, that's the way things are supposed to go. If you were to inject glucose in your bloodstream, which that happens sometimes in experimental or clinical context, you're basically bypassing some of the natural satiety and feeding signals that have been evolved into the body, and your body won't think you've eaten as much as actually you've injected.
Exactly, exactly. And so that's the first thing. They have an incretin effect. All
Mm-hmm.
right. Now park that. Now, gut hormones, of the 20 of them, for example, most of them make you feel full. Of the 20 gut hormones we know about, 18 of them make you feel full. Only two make you feel hungry, right? So in totem, as we eat, as food kind of shuffles through the food to poop tube, shall we say, okay, all these hormones are released, including GLP-1 and GIP. And so when they signal to the brain, they tend to make you feel fuller. Okay. So now that two things, right? So GLP-1 and GIP both enhances insulin secretion and make you feel fuller. Okay. Now then we begin. So it's natural native hormone and its physiological role. And the new drugs are all at the moment modified. They're called incretin based treatments. Some of them are only GLP-1. Many of them are now GLP-1 and GIP as well. But they're called incretin based hormones because the vast majority of these new class of drugs that everyone is talking about are modified versions of
Mhm.
these two incretin hormones.
So GLP-1, it sounds like there must be a lot of GLP-1 sensors in the pancreas, but I imagine there's also some on these neurons in the parts of the hypothalamus we talked about.
Yeah. So the GLP, I mean, there is a debate at the moment, to be fair, about the balance between hypothalamic and hindbrain sensors for GLP-1. Okay, and what the balance is, and I think it will all come out in a wash. There are people who will says it's all hindbrain. There are other people that says that the hypothalamus plays a bigger role, but undoubtedly there are receptors within the hypothalamus and within the hindbrain for these incretin receptors.
Mhm. And so, how does the synthetic GLP-1 weight loss drug differ from endogenous GLP-1 in terms of how it acts, where it acts, and how long it lasts?
Let's start with how long it lasts, because that's its primary difference. The native incretin hormones, GLP-1 and GIP, have very short half-lives. GLP-1, for example, only has a half-life of a couple minutes, 2 minutes. It's then chopped up by an enzyme called DPP, okay? DPP4 in particular, which chops up GLP-1. It's designed to be very volatile, okay? It
Mhm.
goes up quickly after a meal or when you're eating, it then disappears and it comes up again. The the class of drug, these incretin class of drugs, have actually been around a long time, a couple of decades. But for the longest time, it was only a once daily injection. So in other words, through molecular decoration, shall we say, of these specific hormones, they kept it around the blood long enough that you could inject yourself once daily. The big leap came with semaglutide. For those of you who don't know, it's Ozempic, okay, and Wegovy. Whatever reason and how they decorated it, you can ask the companies. These were then once weekly injections. So that's the first thing. The superpower of these new drugs primarily is their half-life, because they stick around the blood for longer. And once weekly is more palatable than injecting yourself once daily. Now, it's a more complex question to answer than you might imagine about what else happens. Okay, but let me try my best to do so. Peripherally, so when I mean peripherally, I mean south of the neck. Um, I think these drugs hit all of the same organs as the native hormone does, broadly speaking. The part of the north of the neck, however, very, very, very little of the native hormone actually probably makes it up to the brain.
Hmm.
Okay. So the complexity is, well, what the hell are the receptors doing there? Well, as it turns out, there are brain specific, what we call PPG neurons, pro-glucagon neurons. So they are the source peptide for GLP-1. There are a small population of glucagon neurons within your brain. And so GLP-1 within the brain is clearly doing some physiological thing, which, would you believe, we don't actually know what? That actually happens in a local brain circuit that is actually there. Okay. It just so happens that because some of these receptors sit, and they probably sense femtomolar, no, I don't know what molar, but sense very, very low
Small
levels of
amounts.
it, very, very small amounts. But the pharmaceuticals that are based on these hormones are hijacking the system to make us feel fuller. So just recap. Okay, peripherally, I think that these new drugs are hitting where the native hormones tend to be hitting. Okay,
Mhm. So
primarily the pancreas.
basically, what you said is, so the weight loss drugs, these synthetic versions of GLP-1, they're engineered to have a longer half-life due to the normal time course and half-life of endogenous natural GLP-1. It's hitting a lot of targets in the periphery, such as the pancreas, after we eat something and it gets down to our gut. But there's probably not much of it that actually makes it up to the brain to stimulate the neurons that are nonetheless sensitive to GLP-1. But because the drugs are engineered by humans to have a longer half-life, they are stimulating GLP-1 sensitive neurons in the brain way, way more than native GLP-1 would.
Exactly, and that's how they work. So therefore, they make you feel fuller. So they're designed to sort of sense all 20 different gut hormones and have a balance of, well, not only how much have you eaten, but what you've eaten as well, how much protein, how much fat, how much carbs. But suddenly, when you then change significantly the concentration of one of these hormones or two of these hormones, depending on which drug we're talking about, suddenly you're going, I'm full. And that is, in effect, the mechanism for the weight loss, because you're full, you eat less, you lose weight.
Interesting. And what's your sort of general take on these drugs in terms of their effectiveness for their primary, marketed use, and what we should look out for or be mindful of as the months and years go by in terms of chronic or long-term effects they might have that we currently don't realize or appreciate?
Okay. So, well, first of all, I've been in this business for 30 years. And we have never had these many effective tools before. And, yes, there's semaglutide, Ozempic slash Wegovy, Mounjaro, which is tirzepatide. Effect sizes anywhere from 15 to 25%. I mean, this a weight loss over a couple of years. Incredible. We never would have thought this possible 5 years ago, but here we are. Okay. So they're very effective. Okay. Extremely effective. They're broadly safe, at least when it comes to the common side effects. And the common side effects, because these are gut hormones, tend to affect the gastrointestinal tract. Okay, so some people may feel a bit pukey, so this side. Some people may have the problem of too fast or too slow in the gastrointestinal tract, so diarrhea or constipation. Those are the three big side effects that are picked up by all the trials. It affects a small minority of people rather than it being affecting everybody, but it does. But that does happen. So we do need to watch it. Well, it's most of the people that start the trials finish the trials, so it can't be all that bad. Now, what you're right in saying is that as more and more millions get these drugs, now we're going to identify rare and super rare side effects. The side effects can be positive or negative,
Yeah,
incidentally.
and to preempt you a little bit here, the trials that have been done, can you give us a sense for about how long those trials tend to last?
So in terms of trials, all of these trials have been 2 years. So
Okay, two
they
years.
tend to 2 years. So they first of all they have phase one, phase two, phase three. But the big phase three trials are all 2 year trials, and they were all completed because no one started dying or anything of that sort. So the safety data for these once weekly, the once daily injections have been around a couple of decades. So from first principles, they are safe, okay, within that context. The problem is when you change from a once daily to a once weekly, you change the kinetic profile, right? So in other
Yeah.
words, different amounts of your. So we know they're safe for 2 years. What we need to know now is that they're safe for 20 years, 30 years, 40 years, and the only way we're going to know this is time.
hmm And the trials that have been done, have those primarily been in adults, like adults? And do you think there's anything worth thinking about here? If, there's definitely a push to give these drugs not only to young adults, but even younger people. Do you think there's anything we should look out for here when we start to think about giving what is essentially a hormone therapy to people who are not fully developed, to broadly speaking?
Yeah, I mean it is a really Pandora's box you're trying to think about here. So now, obviously, the all of the primary phase three, as these drug companies do, tend to focus on adults with the disease in question, obesity or type 2 diabetes. It has been trialed down to adolescents, but definitely post-pubertal. Okay, post-pubertal adolescents, and it works effectively in it. I just get the feeling that for anyone younger than that, it's very tricky, right? It's very difficult to trial these things in children unless they're. So, for example, leptin deficiency, you can be given leptin, but that's because the risks of not getting the leptin to treat the super rare cases of leptin deficiency far outweigh any risks that you're going to get from giving the leptin treatment. Okay, and plus, so there is that. For something like GLP-1, do I see it happening in kids? Maybe, but I just feel that in children, particularly children prepubertal, there are many more levers that we can actually use. We can more easily control the diets of children than we can the moment you get an allowance and you can go spend money on food. Then you're screwed. Then we need to think about other things. But I think for kids, people, understandably, will always be far more careful, particularly when there are other levers to use in order to affect their diet and therefore their health.
Mm-hmm. These are kind of purposely vague questions I'm going to ask you, but given your background, just I would love your general take on. So obviously, we can mess with gut hormones and satiety systems, and it seems to have real effects on how much people actually eat. And then obviously, we've got this obesity and metabolic health epidemic that we're dealing with. So, at a high level, what's your general take on why people can't better regulate their energy intake and their food habits unaided? What is actually driving all of the overeating and obesity from your perspective? And I understand there's probably, you're not going to have the answer, but you probably do have some perspective on this.
I have an answer. It's not the only answer. So
Yeah.
so if we look at so we know of over 1,000 genes, 1,000 loci to be fair. But
Mm-hmm.
anyway, we know of over 1,000 genes that play some role in influencing our feeding behavior and hence our body weight. All right. Now they sit in multiple different pathways and do multiple different things.
Mm-hmm.
But I'll just give you two examples. Okay, so for your brain needs to know. I think I'm repeating myself here, but your brain needs to know long and short-term energy stores, right, in order to influence your food intake. And some of these genes sit within the pathways that sense these long and short-term energy signals. And put simply, and I'm being slightly facetious here, particularly if you're experts in the listeners out there, but in effect, because these some people have brains that are less sensitive to leptin, less sensitive to GLP-1, less sensitive to any of these signals, they are likely to underestimate how much they have actually eaten or how much fat that they're carrying, and therefore make them eat more. So in simple terms, it's like a thermostat, okay? And I think there are many other pathways, but ultimately, what happens is what these genes do in totem is change your drive towards food. Now, this could be reward pathways, hunger, fullness, the full gamish, all right? But some people are going to be more driven towards food than others. And so, given the environment that we're living in, okay, if when you go put gas, when you go buy a Tylenol from the drugstore, and suddenly there's a chocolate bar in front of you, some of us will say and want to buy the chocolate bar. Other people go, ooh, chocolate, and eat the chocolate. Okay, like you're saying that if there's food in your closet, not closet, if you're food in your cupboard, then you're more likely to eat it than if it's not there. Same for me, right? And so that is that tunability, the gene set that drive for food, and there are multiple mechanisms and it's complex, but ultimately it sets the drive for food where some people find it more difficult to say no than others. That is what is happening. So the more difficult you find this, the more aid you're going to need to lose weight. And this is where the drugs come in. Okay. Because some of us can do it through behavioral therapy or group support like Weight Watchers or what have you. Other people will need drugs in order to help you turn down that dial.
To what extent? So when we think about things like just the raw number of calories you eat and whether or not you're in positive energy balance versus what you're eating and metabolic disruption and things like this, to what extent do you think obesity patterns are just due to positive versus negative energy balance and the total number of calories versus the specific types of foods and things that people are consuming? Is obesity merely
So
about energy balance?
it, um, I mean, in one level, it is clearly physics. So it has to be about energy balance. The complexity lies in not the how, which is physics, but in the why. And so ultimately, the output of obesity is a energy imbalance one way or the other. All right, in terms of obesity or being underweight or being average. The complexity lies in the driver. So the complexity lies in the why somebody eats more or eats less, primarily. You might ask, well, what about the other side of the energy balance equation, the metabolism? What's interesting is when we look at these 1,000 genes that influence body weight, they all influence food intake and they all, vast majority, sit in the brain. We have not identified at the population level genes that have to do with your metabolism, which is interesting because twin studies have shown that there definitely are genes to do with your metabolism. Some people are more efficient than others, right? So why haven't we identified them in these big genetic screens? So I think there are two reasons. I think the first is a measurement error, and the second is effect size. So
Hmm.
let's
Hmm.
just I'll break that down. So it's not easy, but it's easier to measure food intake because it's a physical measurement, right? Here's a slice of pizza, here's a can of Coke. We can weigh it, you can look at the volume. The problem with metabolism is there are only two real ways of measuring it, and not even at scale. Okay, you have to either measure heat or you have to measure your CO2 to O2 ratio that you
Hmm.
actually breathe in and breathe out.
Hmm.
So that is possible to do, but very difficult to do at scale. So there's a measurement error. Number two, effect size. Now, a normal size Snickersy, Marsy type chocolate bar is probably what, a couple of hundred calories, 200 to 300 calories. All right. And if I'm motivated, I can probably snaffle down a Mars bar in 30 seconds. But it doesn't matter how fast or slowly I eat the Mars bar, I'll still need to spend 20 minutes on a treadmill or something to burn it off. Okay. So we're designed to eat food a lot quicker than we are to burn it. From an evolutionary perspective, this is what has kept us alive. We're very efficient creatures. So if you look at that, then it is the effect size of metabolism is just far, 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 far smaller than the food intake. It
I see.
doesn't
So,
mean the genes aren't
yeah,
there. It just means it's more difficult to identify
yeah.
the genes.
There's plenty of genes that affect metabolism, and we know what some of them are. But those, what you're saying is those aren't the ones that seem to correlate or map to variation in food intake and obesity.
Correct.
Hmm. When you are thinking about your, so what like, I'll just to the extent that you want to tell us, how has all of your knowledge that you've accumulated and the field that you're in, how has it affected how you think about your own relationship to food? Do you think primarily just in terms of the number of calories you're consuming, or do you pay a lot of attention to the specific types of calories and things like that?
I think more and more, and I've reflected a lot on this actually, in terms of the calorie, and we talk about calories, but they're a unit of energy. And as society, I think we're obsessed with calories. It's a sort of an easy number to think about. People think it's a composite number that reflects the health of your food, how healthy your food is, but it isn't, right? What calories are, what are they? They're a very one-dimensional figure. They tell you what it tells you is how much food you're eating. And that's fine. That's important. It's important. So 200 calories of potato chips is twice the amount of potato chips as 100 calories of potato chips are, for example. But so is 200 grams of carrots, twice the amount of carrots as 100 grams of carrots. And no one is trying to compare 100 grams of carrots to 100 grams of potato chips. So calories give you the amount of food. It doesn't give you anything about the nutrients, the nutritional density of the food. Nothing. Fat, salt, sugar, fiber, nothing. So calories, we need to focus on the nutritional density of food rather than the calories. I mean, for some people need to eat less. I grant you that. But I think a bigger problem we have is nutritional density of food rather than necessarily too much calories.
Mm-hmm. Mm-hmm. One thing I want to circle back to that I intended to get to earlier is leptin resistance. So we started off and we spent some time in this discussion talking about the famous OB mouse and these rare mutations where the body can't sense its fat levels because something's wrong with leptin. Most people don't have that mutation. Most people can have sort of an intact leptin system when they're born. There's nothing genetically defective about it. And yet leptin resistance can and often does develop in people. Can you talk a little bit about what leptin resistance is and what we know about how it develops over the course of the lifetime?
So leptin resistance is an interesting term because it's a controversial term. So I, for one, think that it's a misnomer.
Hmm.
Okay, so first of all, where does this leptin resistance thing comes from, and what is it? Okay, so because leptin reflects fat mass, the more fat you have, the more leptin you have. But when you take leptin and you inject it into a mouse, now it's different in humans, but when you inject it into a mouse will lose weight. Okay, so so leptin is anorexigenic because it signals to pump C, and you end up eating a lot less. But where the leptin resistance comes from, but wait a minute, but the fatter you are, the more fat you have, and the more fat you have, the more leptin you have. Why is the leptin not signaling to the brain to let you know that you have now too much fat? Please start eating less. That is the leptin resistance. I think it's a misnomer. And I think it's a misnomer because it's a pharmacology question
Mm,
rather than a biological
I see.
question.
So the raw observation is there's lots of people out there with high levels of fat and therefore high circulating leptin, but their body doesn't seem to be responding to that in the way you would expect. But I think what you're about to tell us is that may not be resistance at the level of like what a pharmacologist would say.
Exactly, exactly. I think it's where if you have pharmacology, for those of you who remember your basic pharmacology, you have a curve, right?
Mm-hmm.
And you obviously have where does the curve meet that sort of the maximum effect size, and where do you actually saturate the system and have no longer any effect? I think leptin is designed to function very, very low concentrations because
Mm,
it's designed to turn on the starvation response. It's designed to tell you
yes.
you don't have enough leptin. And so when there's loads of it around, which means that you actually have enough fat, your brain just
Mm,
says there's enough. It doesn't matter whether or not you have 2 kilos of fat, 20 kilos of fat, or
yeah.
200 kilos of
The
fat.
system
You have
didn't
enough
evolve
fat.
to sense that because that much fat was never really an issue.
Exactly.
That makes
Whereas
sense.
it is sensed that really tight thing
Yeah.
between
Yeah.
200 grams of fat to a kilo of fat, something like that.
Hmm, hmm. Okay, that makes a lot of sense actually. Okay. So you don't okay, so it's not I see what you're saying. It's not when we talk about resistance, this is not what we would think of as classic like drug resistance or something like that. Like the receptors have been internalized on purpose because of something some fine-tuned effect of having so much of something floating around.
But that being said, clearly, if you can find out the mechanisms, okay, that would enhance leptin signaling. Could you, for example, improve the sensitivity or change that within your brain so that you could respond to additional leptin, or your brain becomes more responsive to a broader range of leptin?
Yeah.
And so there are companies out there with drug programs that are trying to do that. They're trying to sensitize your brain to leptin so that it can be used, probably not in isolation, probably together with GLP-1s or something else, to actually increase the effect size of some of these drugs. So while the concept of resistance, to my mind at least, is a pharmacological question rather than anything else, it doesn't mean that it doesn't mean that we can't at least learn from it to try and design new therapies.
Hmm. I see. I see. Well, we've discussed a broad range of topics related to the genetics and neurobiology of feeding and hunger and obesity. What are you working on nowadays in your lab related to these topics? What are the exciting questions that are being asked that you think we're going to start to get answers to in the near future?
So, I mean, there are two things that we still need to understand more about the genetics. In other words, because ultimately, I think what we are beginning to realize is, as I said, obesity is a simple output of physics. But because each of the drives, each of us have different reasons why we end up eating more, we need to understand more about these. Because the more about this we understand, the better we're going to be at beginning to tweak the system. And so we're still trying to identify new genes, even though they're super rare. Okay, super rare now. Nothing that we're finding is particularly common. It gives us new pathways to begin to say, well, are these tweakable pathways or not? Most of them are probably not going to be tweakable, but some of them will be. So that's first one thing. So we're working on still looking at rare genetic causes of severe obesity. That's one. But second, and the far larger program I'm working on at the moment. Now, because feeding circuits are circuits, they tend to be within the brain. The vast majority of our understanding of feeding circuits have come from mice for obvious reasons. Now, however, and given that we're chucking drugs at the brain for all kinds of things, shouldn't we know what happens in the human context? So now we have access to brain donor samples, and the techniques have now matured to the point where you can now do single cell and spatial approaches within donor brain samples from archives. So not even like fresh donor brain samples, from frozen samples. That's what we're doing. We're now mapping feeding circuits. We've just, for example, produced a spatial map of the human hypothalamus, the normal weight human hypothalamus. We just published that 2 months ago. And we're now looking at that in lean and obesity, and we're doing the same thing within the hindbrain as well. So I've become a glorified cartographer. I'm trying to make maps, but within the human context.
Mm-hmm. Is there anything else you want to say about what we've discussed, or any final thoughts you want to leave people with about the broad topic of obesity and feeding?
No, I mean, I guess one final comment, because we did discuss these drugs for a bit. I mean, something that people have asked me is, with these drugs, have we solved obesity? Is it almost a panacea? And I'm going to say no. And I'm going to say no for a couple of different reasons, because I think these drugs, not everybody responds to them, but a lot of them do. That they're designed to treat obesity, and I think we do need to do so, but they're not designed to prevent obesity. So prevention of obesity still needs to fix the food environment we live in. All right. And that will require policy, legislation, and a bunch of levers that drugs are not designed to pull. The second is these drugs are powerful and they'll make you eat less, but they won't change what you eat. And so we still need to find ways of improving our diet as well, in addition to just taking the drug. So I think, just as a takeaway, just in case people think I'm this drug shill, which I'm not, okay? But anyway, I think these drugs are a tool, but it needs to be within the context of broader societal changes. And we still need to eat healthily, even when we're eating less.
Mm-hmm. And I guess, tell me if you would say this, but it sounds like you would probably agree with the statement that whether or not you're just using your own willpower or using one of these drugs, if you're somehow eating less, you still need to think about what you're eating, because in my view, the only way the food environment will actually change is if consumer demand changes. So until people start, voting with their dollars and actually eating different things, it's not, the food environment's just not going to magically fix itself.
It can be fixed through policy changes if the governments are able to do so. You know what the big problem is? The big problem is that there is always this tension between public health and the loss of personal liberties. Okay, we drive on a specific side of the road. We stop when the red light goes red, and we wear seatbelts. Okay, so we can't do whatever the hell we want. So and reason why is because it keeps us safe. Now the problem is this, right? So in other words, for public health purposes, how much liberties are we willing to lose? Now this line is murky and gray, never politically expedient, but it's going to differ between society to society, and culture to culture. But we need to reach this balance of, well, how much personal liberties am I willing to lose to have a healthier society? And that needs to be accepted by the majority of society. And so, yes, I think that the food environment, that is the only way we're going to tackle it, through the loss of certain liberties. I'm not a nanny state person, please, but that's the only way we're going to be able to do this societally.
How does that dialogue happen? So I mean, it's I can think about this abstractly in the idealized case, but the fact is we live in a world where Nestle and Coca-Cola and other interests basically control the food supply almost top to bottom, and like it or not, they are able to integrate themselves into the policy apparatus in a way that circumvents the dialogue between the people and the policy apparatus. And so how do you think about that?
No, and you're absolutely right. And I'm not saying that any of this is a quick fix. So we need, I am one, and I am a believer of engaging with these companies, Nestle, Coca-Cola, et cetera, et cetera. Okay. Some people say we shouldn't talk to them. It's like trying to talk to the cigarette manufacturers. There's a difference between cigarette manufacturers and food manufacturers in that we don't need cigarettes, but we need food. So I do think we need to engage with the companies somehow and get them. How would I think about this? I think that we need to try, naively perhaps, we need to try and convince the companies that if they make healthier options, that we will live longer to spend more money with them. So if we make healthy food profitable, and convince the companies this is so, then market forces will make the company actually change that. So in other words, I think that the bottom-up approach, so in other words, the consumer voting with their feet, is never going to be this only solution, because the problem is we have chimp brains. And when are we going to vote for chips in English, French fries, against broccoli? We just won't do that because we've got French fries. Chimp brains, all right? And so what we need to do is we need to have companies as well understand from a marketing perspective that, can we make healthy foods tasty? We probably could. And can we make this profitable? Mm-hmm. And they probably could as well. And so you're right. It is in no way am I saying this is an easy solution, but I'm saying that we at least need to have the discussion.
Yeah, yeah. I understand what you're saying conceptually. If you could convince and it sounds very logical from one view.
But naive.
Yeah, I mean, if you can convince the companies, hey, if the population is healthier, they will live longer to spend more dollars with you over their longer lifespan. That works unless these companies directly or indirectly are able to profit literally from the other side of things, which is chronic disease. So if you're simultaneously profiting from the foods that make people chronically ill and from what they have to do in order to treat their chronic illnesses, then that argument would go away.
You're a cynic. I sense this.
I'm not sure I would agree with that, but such entanglements do exist. And so I'm just saying your argument isn't going to make them go, oh yes, let's just make healthier food because
no. Otherwise it would have done it. If my argument works, it would have already solved it. So clearly this is living in a slightly idealized world, but we need to sort of conceptualize what is the framework within we can begin these discussions, I think.
All right, Professor, we've covered a lot. Thank you very much for your time. Very interesting stuff. Professor Yeo, thank you.
Thank you for having me.
Visit mindandmatter.substack.com to find all of my content.